It's a great pleasure and honor to introduce Crystal Cooper as our next speaker. I know Crystal for almost 5 years now. I still remember when I hired her here at Cook Children's, like that it was COVID, so we couldn't even shake hands, and we were wearing masks. We work very closely with Crystal. She's doing a phenomenal job on different projects regarding comorbidities in children with epilepsy. Crystal is currently an assistant professor of psychology at UT Arlington and principal investigator here in neuroscience research. She received her doctoral degree in health psychology and neuroscience at UT Arlington, where she specialized in cognitive neuroscience. She went to UT Southwestern Medical Center for her postdoctoral research fellowship and early faculty career in psychiatry. She has nearly 50 publications, as well as numerous conference articles. She, as her trainees, have received awards for her work. She serves actively as an ad hoc reviewer for several journals, and she's a member in national and international scientific societies. So Crystal investigates biosignatures of neuropsychiatric comorbidities using measures of behavioral and brain function. And today she will present her work. Please welcome Dr. Crystal Cooper.
Thank you, Christos. To follow up from Dr. Bowie's panel, my daughter just won district at her science fair for 5th grade. However, I was quickly saddened to hear that for career day on Friday, she did not want to go as a scientist because she was like, what do I wear? I'm like, well, I have lab coats, go as a scientist. She's like, Mom, I know I won district, but I don't want to be a scientist. I want to be a YouTuber. I'm like, oh no. So anyways, children are interesting. But in line with that, I hope my mentees, which you'll hear a lot of their work from UT Arlington, as well as some fellows that I have from JPS and medical students from TCU, hopefully, they will go on and do great science or facilitate science in their medical career. So I'm going to discuss 3 things. There's 3 main objectives here. One, I do want to talk about concepts of brain and behavior and frameworks that shape the way I do science. I want to talk about multimodal data techniques for investigating brain and behavior, and also highlight the importance of partnerships in this neuropsychiatric work that I do, because I do work in patient populations, so that cannot be done without patients. So I want to take you through a little bit of a journey of how I got where I am. So UT Arlington is my alma mater. I got my PhD there in health psychology and neuroscience, where I studied cognitive neuroscience. But that was not initially what I wanted to do. I wanted to go to medical school. I was a bio major first, and I happened to I took research hours in the EEG lab, and of course I fell in love with looking into the brain. But Ronald Reagan passed away during that time, and a lot of information was now just flowing everywhere about Alzheimer's disease, basically the brain-the-mind connection, and I fell in love with that. And I said, I want to be part of the unknown and know more. Now, since then, we know a lot more about Alzheimer's disease. So I switched to wanting to study the brain and the mind, and so I did cognitive neuroscience. And once I finished my PhD, I went to UT Southwestern in the Department of Psychiatry to study depression and anxiety in this sort of biological psychiatry framework. So the tools that I use to study the mind and its neurocorrelates, we were using to try to identify some true, hopefully biosignatures of depression. Because before that, and even now, what's the main way that we diagnose depression and anxiety? We ask questions to the patient, and those will always be needed, but it's also nice to know, is there anything about their biology and their behavior that can help us to both better subtype them, predict their treatment course, and what to give them early on. But in my time at UT Southwestern, a couple things happened, and a few key moments for me that sort of hit me as a scientist that got me into the field that I'm doing now and doing work with Christos and Scott and the team here at Cook and our trainees at UTA. I was at the MRI scanning a kid. He was about 11 years old, and I had assumed he was a healthy control, so we have these nomenclatures for naming things because nobody uses the name. And I started putting basically healthy control. And my research assistant stopped me and said, no, he has depression. And my son was just a few years younger than that. He was my firstborn, and this kid was only a few years older than my son, and he had depression. And you wouldn't have known when you met him, and it just devastated me. The second thing that happened was I was helping oversee brain and behavior of all the studies done at the center we were at, but there was this big study we were starting, which is a natural history trial, which was to look at risk and resilience against depression from early age into adulthood and understand the difference between those that end up with mental health issues and those that don't. And a lot of people were coming in with comorbid neurological disorders, like kids with epilepsy, moms with MS, and they were coming to me like, Crystal, how do we handle these? Because before in clinical trials, you automatically exclude people like that. It causes noise in the data. And I was like, well, we can treat them as something separate. And we were starting to look at the numbers and realizing how many of our cohort had these comorbid neurological disorders, with one of the most high ones being epilepsy. So I got interested in this sort of like topic of comorbidities because it's messy. No one really wants to touch it. And COVID-19 or COVID was happening. And I interviewed with Christos, and specifically about this passion of doing this combined work. And he said it's perfect. And I also interviewed with Scott. He's like, you speak a language that some people don't speak, and in science and psychiatry. And so we felt like it was a perfect fit. I wanted to really dive into the sides of comorbidities in this neuropsychiatric field, and they were interested in expanding the neuroscience work. So Christos is like, you're great. He and I are completely different from each other, both probably in personality and how we look at the brain, and I think that's important. I see the brain different than he does, and our work is complementary in that way, and I'll get to talk about some of that. Now, since my time here at Cook, we've had partnerships with UTA, mainly bioengineering initially, and now I've come on board at the Department of Psychology, just starting this academic year, and so really one of the first to start this joint appointment process between Cook and UTA, which Dr. Wydance will talk about more later, and then you'll hear from Dr. Marshall tomorrow about some of the work that Cook does with TCU, which I'll highlight a little bit about that too. So I do cognitive neuroscience. For those of you that don't know, that's essentially the subspecialty of trying to identify the biological substrates of cognition, so mental processes. With advancements in technology, we've learned a lot about how the brain is functionally structured, or functionally divided, not just structurally divided. Specifically, in the most simple sense, like 7 major networks. Now, these can be subdivided into other networks. Now, prior to technology advances, how did we know what happened with people in the brain, and like how that manifested behaviorally was through lesion studies, like Broca's, right? Can't produce language if this region in the brain is not, there's a lesion there called Broca's now. Same thing with HM. He had epilepsy. They went and surgically removed his hippocampus, and we quickly found out that you need the hippocampus to form long-term memories. So through lesion studies, we've learned a lot, but now with advancements in MRI, EEG, MEG, we see a lot more about how the brain is functionally divided up. And that is what I'm interested in, is how the brain works functionally. And so Christos already talked about these, so I won't spend too much time on the differences between MRI and MEG, EEG. But I will say that generally speaking, I want to just discuss the difference between both. I've actually borrowed this and modified it from some of Christos' talks. MRI is great spatially. You can get down to the millimeter in terms of spatial resolution. Temporally, not great when you're wanting to look at functional data. It's like across minutes rather than like seconds, because it's based on a blood response. And so you have to follow this blood response. That's obviously not as immediate as neuronal firing, because the brain can't store its own oxygen or anything like that, so it has to pull it from the blood. So when your brain's active, you see this change in oxygen in the blood, and that's how we know aproxia, okay, that area is activated because there's a drop in oxygen, put simply. Now, MEG, EEG, sort of the opposite. You have this great temporal resolution. Like neurons fire, they cause these electrical and magnetic field changes, and you can immediately measure that with these electrodes. Spatially, not as great as MRI, and so what obviously Christos and his team's work is really trying to identify how to make it more spatially high resolution. But it's important for us to combine these in the work that I do. So there's 2 ways to kind of interrogate brain function, and that is at the resting state. So just at an idle state, they're awake, just thinking to themselves, and you can look at how the networks are connected at the resting state. You can also look at the active state. So you give them a cognitive task, they're challenged, the brain is, responding to that, and you're able to actually measure what's happening in the brain at the time of this cognitive task. Now, I'm going to highlight a couple different cognitive processes that I've used through the years to interrogate different things. So, and I've divided them by non-effective and effective processing, and there's a reason for that that will become more clear in a second. So non-effective mean that it's just what we call like kind of cold cognition. There's no emotional or hedonic component to it. So if you want to probe hippocampus or temporal lobe, you can do a binding task for face name memory. I have you study these face name pairs. I only show you the face later, either ask you to remember the name, or I show you a mixed up version. You see if you can detect whether that was a pair that you saw before. I can also probe frontal lobe function with like an inhibitory task, like the flanker task, where your job as fast as you can is to respond to what direction the middle arrow is facing. So those flanked arrows sort of confuse you. And so it's sort of this inhibitory response that you have to control in order to give the right response. Now on the other side, you'll see affective processing, which means we sort of layered in an emotional component or a hedonic component. So in this emotional conflict task, it's sort of like a Stroop, the color word. Some of you might know color word interference task, where you have to say the color of the ink rather than reading the color of the word, which is actually really hard for us to do because both of those come naturally, but we read really fast. In this case, we all know how to recognize faces, and it is in our best interest to understand if somebody has a fearful expression on their face or a happy expression on their face, so we know how to respond in the environment. We also know how to read, and we process words really fast. So the word fear, you know what that means, and you process that quickly. So if I'm having you go through this task, I'm like, you need to respond to the facial expression and try to ignore the word. There is a conflict layer there that you have to regulate in order to give me the right response. On the other task, I can give you a task where I'm just having you press levers to different stimuli and you don't know, but I'm giving you more positive feedback for one of your button presses and more money for that button press in hopes that you will implicitly start pressing that more. We are animals, right? And so it's a lever press game. And the idea is you can learn from rewards and you can provide a response that is a biased response towards the reward. So hedonic experience. Feels good to win money. So you want to win more money, so you're going to hit that button. So that's what we like to look at in terms of reward response. I bring that up because in the world of depression, we can't ignore the emotional and hedonic components of everyday life and how that kind of folds in with our regular cognitive processes. Depression, about 1 in 10 people have depression. This is across most age ranges, of course, I'm overgeneralizing there. But one in every 11 seconds there's a suicide. And for people between the ages of 10 and 24, it is the leading cause of death is suicide. Depression is huge. And I learned, as long as I've been here, epilepsy is huge. You have one of the #1 neurological disorders, one of the #1 psychiatric disorders, and they exist together in great numbers. And we need to understand that comorbidity. But before I get into that, there's 2 ways in which we measure depression. So one is through clinical diagnosis, through structured clinical interview. But then there's also ways to track treatment response or to understand where somebody is in a current state through symptom severity checklist. And so they go and they answer these things like the PHQ-9. So there's 2 frameworks that sort of kind of motivate the way I do research. One of them is in the world of biological psychiatry, where NIH built this framework to look at sort of domains of the human condition. So cognitive processing, positive, negative valence. How do we measure those things with different units of analysis like brain imaging or genetics or whatever across the lifespan, different environments. But the idea is that most symptoms that we think happen in psychopathology happen because there's deficits in cognitive processes, and most of those have a neurocorrelate that can be identified. Similarly, in the world of neuropsychology, there's this idea of understanding hot versus cold cognitions, that in psychopathology you have this sort of like blunted reward learning, this overactive emotion processing, and sort of blunted or executive function. So like your top-down processing is sort of not working well and your bottom-up is overexcited. So I like to bring in that framework into different modalities. So I'm sort of like a walking multidisciplinary like researcher, where I bring in the field of like neuroscience with the field of cognition and the field of like psychiatry. So I bring in clinical assessments of mental health, brain assessments using different imaging techniques, as well as different types of tasks to probe certain functions that we think might be the problem in a particular condition, like epilepsy comorbidities. So now I'm going to switch to talk specifically about some work that we've been doing here at Cook in the world of this neuropsychiatric dysfunction. So Christos' Neuroscience Research Center and Scott, who's the head of neurosciences, who brought him on. Like I said, I interviewed with them, and I quickly realized when I was walking through the Cook facilities, they have everything that an Ivy League university has, and they have also the physicians that want to help do the research. With the caveat of, this is a hospital. Like, the work that we do as scientists have to also fold in pretty well with the way they do clinical care, and they're more than happy to help you. And they care about the work that I'm doing. I've also talked with Dr. Shahani, who, part of the work I do, I'm not presenting here, is in psychogenic seizures. And what I've learned in the field is that, in depression we actually just exclude people with, neurological disorders. But in the world of like neurology and understanding epilepsy, it's not that they ignore depression or exclude it. It's just their goal is to get the kids seizure free, and they know that sometimes there's these other issues, and they try their best to help there. But most of the time, by the time it appears, they've been struggling with it for a while, and it can be a problem. It's harder to get them in remission and keep them in remission longer by the time the depression has gotten to a point where it's clinically measurable. So I'm going to highlight some sort of different areas of this neuropsychiatric work that I do using the work that my students at UT Arlington are doing, both in bioengineering and in psychology and neuroscience, as well as some of my undergraduate McNair scholars, and also a TCU medical student and some of our JPS child and adolescent psychiatry fellows. So now, specifically I'm going to talk about the work that our students in bioengineering are doing and the work that my students in psychology are doing. So as I mentioned before, depression is already a bad thing, and what I want to highlight here is in the general population, this is sort of, where is our mouse? There we go, okay. There we go. So here in the general population, you can see that this is sort of the rate of psychopathology, but within epilepsy, it's nearly half of them will have some sort of depression or anxiety disorder. So, and it's not just that it exists in epilepsy. If you have depression and or anxiety, particularly depression, your chances of having a seizure in your life is increased. So it's not just one way that you have epilepsy, and so therefore you might have depression. When you have depression, you actually increase your risks of having a seizure in your life. So in coming here, I took what I've been doing in the MRI world, because most of my experience was in fMRI, and studying biological psychiatry was more in the space world of MRI, less so in the time world, like EEG, MEG. And basically moving what we've been doing in MRI for depression and moving that into epilepsy in the MEG. So Christos and his team had to help train me in MEG so I could learn that. So that we can understand not just like where stuff is occurring, but at what time. Because it's important to know when are certain networks coming on and offline. Epilepsy is a time disorder, not just a space disorder, right? You have to understand how its brain is working across time. So I taken one study that was funded by the Jordan Elizabeth Harris Foundation, was one of my pilot works, brought in kids who were pretty well controlled of their seizures, but we wanted to understand how is their brains working in terms of mental health and cognitive function, and what are the neurocorrelates of that. So I brought in all these different techniques into this population, and they were recruited here from our clinic. So the work from Catherine King, my first PhD student who came from bioengineering, took this task that's been done for a while in psychiatry to understand it. It's been great at both identifying pathology differences, like subtype differences in depression and anxiety, but also predicting treatment response. So we know what to expect in terms of brain regions, like amygdala to activate during conflict, and then you have sort of ACC for implicit regulation, the DLPFC for decision making. So we have this general idea of what's supposed to happen, but we've never watched it happen in time. And what she was able to show was it happening in time. So this is a healthy control paper that she published that shows that we can use the same task that's been used in biological psychiatry in MEG for healthy controls, and I'll show you the epilepsy in a second, and see when the network is coming online and offline. Like, I've never seen the amygdala go on and then off. So that's not what you see in fMRI. And then here, when she did the comparisons between those with epilepsy and the healthy controls, we saw all these deficits. Blue means deficit. So we saw the deficits in the network and in time. And we were able to identify that using a task that's been pretty successful in biological psychiatry, we actually can show deficits in just the basic processing of this task, and those brain regions, that activity was related to their depression and anxiety levels. It predicted their depression and anxiety levels. So it's not just that it was a deficit across the board, but that it specifically was predicting depression and anxiety at certain time points. And she just started this work on connectivity, because I like to see the brain as a network, and as networks turn on and turn off, it's like a car. You can't drive forward and backwards and be in park at the same time. The brain is like that. It's moving, and the networks are dancing. And she actually won an award at our conference at AES, the Grass Young Investigator Award. So the work's out there. Go see it in the poster session. All the students I'm showing stuff out, go see their posters. They're here. They'll give you more information. But what she's been able to see is that she was actually comparing two types of analytic techniques, which I won't go into the specifics, but effective connectivity versus functional connectivity. Effective connectivity is essentially, she's using task and directional type connectivity, so strength in and out of certain regions, between certain regions, and she found overall that you see the blue lines, healthy controls generally had greater connectivity than epilepsy, but it was not always the case. There were some cases where epilepsy had greater connectivity than healthy controls. Now, why is this important? Well, based on the areas where it's default mode network, which is our resting state network, salience network, or executive function network, if we can identify where they might have disrupted connectivity, can we target those? And or would it be, one reason I did the task that I did, which I didn't mention before, the emotion conflict task, people ask, why do you do that? I was like, well, it crosses subcortical and cortical regions. It basically engages the whole brain and a lot of serotonergic-based systems. And like reward tasks really promote dopaminergic systems, so it depends on what system you're wanting to interrogate, but this sort of emotion conflict brings online the whole brain. And so she was looking at effective connectivity of that task and how is the brain as a network connected during a task like this, and we're able to find differences in these major networks that we think are involved in psychopathology, and so how can we target those for not just their epilepsy, but for their depression. And I'd also like to highlight some behavioral work that we've been doing. So outside of the scanner, even though like in the MEG I did this particular task, I do a whole battery of tasks outside the scanner so that we can identify behavioral deficits and then maybe correlate that to brain data after the fact. And so one of my other students, who I co-mentor with another faculty member at UT Arlington, was really trying to compare two types of like control, both the flanker, which is a non-affective control process, and then you have an effective control process with the emotion task. And she was able to find with the with this interference task here that we had people with epilepsy had a greater interference effect, which means that they showed greater interference that they had to then regulate based on their performance. And not just that, it was related to their anhedonia symptom measures. So we have these measures of anhedonia or hedonic experience, like how much you take pleasure in life. Do you like walks on the beach, or do you like eating pizza? Things like that. And a high score is good on this measure, the more normal your hedonic experience is. So a low number is bad. So what we found was the greater the interference effect, the lower their hedonic scores were. So they're more anhedonic. And in fact, that they actually had a higher PHQ-9 scores, so higher depression rates. I also want to highlight work that I initially started with a McNair scholar of mine looking at reward processing, because we do believe that reward learning is a huge problem in depression. And so we wondered, is there a problem with it in people with epilepsy? Do they struggle with learning from rewards, and therefore could that be one of the symptoms that they have, is this anhedonia? And initially, we did not see a difference in reward bias, which is what I told you about that behavior, like I'm going to reward you more, so therefore you should hopefully start to click that lever more. We didn't initially see a difference there, but they were much slower at responding to the task, so we think that there's more going on in their brain to get the same job done. But we weren't able to identify a behavioral deficit, but doesn't mean there's not a neurological difference. And I've seen that in past work that I've done where two people might perform the same on a task, but their brains are doing vastly different things to get the job done. You see that in like aging work. And also accuracy on this task also was related to depression. Now, my current PhD student, who has a master's from Tarleton and is working on a PhD with me, is wanting to be in the field of cognitive neuroscience. And we're taking this diffusion drift rate model, which is a fancy model of doing math for decision making. And the idea is, you know, response bias, as we've traditionally measured it, is through signal detection theory. There's other decision-making models out there, some are economic, but in this one, drift diffusion, the idea is, can someone with epilepsy, they're learning, but are they learning at a different rate? Does that make sense? So like maybe healthy controls learn at a faster rate than those with epilepsy. So they're all learning, and they're doing the job, but are they learning at the same rate? And that's what we're trying to identify, and then take that drift rate model and put that into the brain to identify, is this related to how their brain is functioning? Is that why they're slower at doing the task, but they're learning, and can we identify what's going on in their network? So now I'd like to highlight some resting state data. So this is MRI data that we've collected on a scanner here at Cook. Florencio is another McNair scholar of mine. His work is also out, so all the students' work is out in the poster area. So in the field of psychopathology, there's this theory out there that all depression, anxiety is probably stemming from issues in this triple network model. So it's this model of the default mode network, salience network, and executive control network, which can then be subdivided into this dorsal attention network and this frontal parietal network. So there's we take for granted everything that our brain does, but these networks work together. Some of them need to turn off, and some of them need to turn on, and some of them have to work together to get the job done. And so there's this theory that all psychopathology is probably stemming from an issue in this triple network model. So let's look at it in epilepsy and depression. And what we found is that there were connectivity differences such that those with epilepsy had this greater connectivity of their default mode network to like the salience network. And there were some disruptions in their salience network and this frontal parietal network. So kind of highlighting, this is getting into when we control for the role of depression, you see this difference in their connectivity. I'll explain the sphere for you here. So here I have it divided by default mode, salience, and executive control. So that's what the sphere is. So you can see how everything's connected. Colors-wise, red means higher connectivity of the first group than the second group. Blue is higher connectivity from the second group to the first group. And so the idea is we can see sort of connectivity difference visually, and then we depict them in the brain, like so you can see them cross the frontal to parietal lobes. And what we found was we think there's this disruption potentially between internally and externally driven attention, and this might also stem into their ability, what I talked about before in a framework, is to have this top-down control or this overactive bottom-up processing. There's a lot more work to done in the resting state and hopefully kind of related to some of the cognitive stuff that we have. And we have a couple of my students are also working the MEG, EEG, doing the same thing as this. And I also want to take a second to talk about work I've been doing for the past couple years, partnering with TCU and JPS. Specifically, and you'll hear from Dr. Marshall tomorrow, he sort of spearheaded, bringing in TCU medical students to do research here. Part of their program requires them to do research. And so we bring them on. Christos has had plenty of TCU students, and I've had some TCU students as well. And then about 2 years ago, Scott and another professor, a psychiatrist at JPS, wanted to open up a psychiatry fellowship opportunity where those psychiatrists that wanted to do a fellowship in child and adolescent psychiatry would have an opportunity at Cook. Because at JPS, you can't guarantee you're going to get experience doing research or even seeing a lot of kids. And so they worked hard to build this partnership. And so there was work that I've been doing to sort of take these TCU students and these the medical students, these JPS fellows, and have them do work in a field that I'm passionate about, that Scott's passionate about, and Christos is passionate about. And so kind of harnessing a database, which we'll talk about in a second, and then hopefully the next steps with merging the work that I'm doing and Christos is doing to bring a bigger picture of the behavior and brain deficits in kids with drug-resistant epilepsy. Like I said, their goal is for 2 years they're working for us, they have an output, right? So a poster or paper, things like that, and their posters are here. The work's also been presented at the American Academy of child and adolescent psychiatry. So I'm going to talk first about what we've been doing. So there's this large consortium called the Pediatric Epilepsy Research Consortium, PERC is the initials, and Scott oversees that. And there is this, the kids come into our epilepsy monitoring unit, those who have drug-resistant epilepsy, and they're getting evaluated based on standard of care. They're getting everything done. What we noticed wasn't happening very regularly is they weren't getting evaluated for depression or anxiety often enough. In fact, it was probably about 3% of the time, based on probably the clinician thinking there could be a concern. So we decided as bringing on these fellows, and what I want to do is if we can get more of that in the database and more of that information in the system, then we can take that information, not just help them, one, get caught early and get help. We can also look at hopefully the neurocorrelates and the other behavioral correlates related to the psychopathology and understand how that might impact what we know about brain and behavior and also prediction of treatment outcome. So as part of this endeavor, we passed out these packets that are self-reports. They report on their mood and anxiety and even ADHD, an executive function score, and a pediatric quality of life questionnaire. And so they do these just part of their EMU stay now, and the team looks at that, the trainees look at that and make sure there's no critical item like suicidality, and if that's the case, they call someone while they're still in the EMU to go see them. If they have high levels of depression or anxiety or ADHD, the clinician follows up with them or social work follows up with them. But here's what we found in looking at this data. We started it in January 2024, that they started passing out the packets and looking at that data. And what we found was, if you look at this bar graph here, so we have depression, anxiety, we have hyperactivity and attention issues, so those derive the composite of ADHD. And so you can see within our drug-resistant epilepsy, we're seeing the rates that we sort of anticipated to see, but we were not capturing as much prior. So now with every kid coming in, we've only missed about a handful of kids due to short stay or just not filling it out. And we were able to then hopefully get them, like I said, followed up with care. So Marcia Gabriel is one of the neuropsychologists that I've been working with, and she debriefs the kids when they score high on this stuff. But more importantly, what's something that Rumsha was specifically interested, as well as Allie, was looking at, well, how do these things relate to quality of life? And so we found that their levels of depression and anxiety, as you might expect, were negatively related to quality of life. So the more depression symptomatology that they had and anxiety and issues with attention and hyperactivity, the poorer their quality of life was. Now, Mariah, she wanted to look at something like I said, I always go back to the cognitive side. Like, I care about the mood and I care about the psychopathology, but I also care about what's the cognitive process that might underlie why we see what we see, so we can then look at the neurocorrelates of it. So she took this executive function questionnaire and said, okay, well, let's look at different types of executive function. That's a very broad term. We do a lot of things to problem solve, plan, emotionally control, and things like that. But there's these subscales. And what we found was that emotion conflict was related to different symptoms of psychopathology, and then also planning and organizing. So for instance, emotion conflict, deficits in that were related to depression scores. And then also what we found was anxiety was related to issues in planning and organizing. And if we divided up for those that had normal numbers of these scales versus high, we actually did find that they were significantly different in what their rating was for depression and anxiety. Now listen, why is that important? Like, this is all behavioral, like we can measure it in neuropsychiatric ways and also cognitive ways, but like I said, I always go back to I want to identify also its neurocorrelates. So that's the next step. Our next step is to look at the neurocorrelates of this function, and then hopefully identify pre or post-operatively, does this help us understand how well they're going to do later? Now you might ask, like, well, how does this matter outside of even drug-resistant epilepsy? So like, Christos and I were talking, actually we talked about just a couple weeks ago, it's like, okay, how can we look at what he's been doing in the drug-resistant epilepsy with what I'm passionate about and what I've been working with Scott and Marcia with on trying to get all these kiddos screened. How can we bring all that together for kind of this more holistic care? And so we have this idea, again, to bring in the triple network model and say, okay, let's look at these certain brain networks, combine it with what he's been doing, and then us being able to get this information now into our database about their psychopathology to better understand and hopefully treat predict treatment response. But ideally, it goes beyond that. So like if we have kids coming in, and we can identify through some tests. Now, again, I'm not necessarily saying that we're going to do tasks all the time to get at this. That's why resting state is so important. But if we can identify, so this precision medicine model, we have all these first-line treatments, second-line treatments, and they're very specific. But if we know that somebody is at high risk for depression, anxiety, like with their epilepsy, and they have a certain signature, certain like brain pattern, like biosignature, can we then be more targeted with augmented therapy? Like, different types of, whether it's drugs, whether it's other types of like behavioral therapy. Is there different ways that we can target to help them get better and stay better longer, even hopefully before they actually have like onset of significant symptoms of depression and anxiety? With that said, I do want to summarize, not to be too cheesy about connecting the dots, but importantly, like we care about getting care for the kids, doing better science for the kids, and it takes a team. Like it takes doctors like Scott, it takes scientists like Christos, myself, it takes other clinicians like Marcia, and then students. This is just a summary of the people. I understand I'm not trying to just highlight them, but we can't do it without the team and all the institutions now that we're teaming up with to do better science, like UTA and TCU and JPS. The whole idea is even though our PhD students at UTA are going to go off and do science and maybe they'll stay in the clinical world, and we hope that, but even physicians coming in to do rotations with us, we want them to be more passionate about research and be involved. Because not every physician wants to do research, but I've noticed at Cook, most are willing to, and that's what's beautiful, and hopefully we're training up the next people to do the same. So I'd like to acknowledge the whole team. What's a beautiful picture here is there's actually students from all those institutions pictured here, with most of them being from UTA. And so, of course, Christos, thank you for, taking the risk with me. And this is all very exciting, these new endeavors that we get to take together. That's a QR code if you want to go look at our website, sign up for some studies. And do you have any questions?
Hello.
Hi.
I'm Allison with but I work in stroke and thrombosis endowed chair. Have you looked at applying this to patients that have severe neurologic deficits or have suffered a stroke? Because I'm wondering if they have that severe damage, if that would work at all, or if you've even considered that.
No, I think it's a great area. So obviously, Christos has worked with different types of, I don't like to use it with brain damage, but impact and how that matters. Like, yes, I've actually talked to a couple different people what that could look like the kind of work that we could do there. And what's nice about sort of partnering with these different institutions is you can't do science without a team. Like, you need a team of scientists, and you need obviously the hospital partner. And this is an area that I'd be super interested in, just haven't had the manpower to do it. But I think that it's definitely something worth considering, because based on area of impact, it's going to change networks and it's going to change systems, and we need to know how to target those to help them to have better daily life function. So I think it's a great area to research, and they're also at higher risks of psychopathology after impact. So, all of that matters, all of it works together, which is why it's so messy, but it's important to look at. And so it's an area I would love to look at, and it's something that Christos and I have talked about, and some others. Just you need the team to do it.
Right.
So, and yeah, funding's also great. You have funding to do it. So,
Well, thank you for everything you're doing. It's great.
That was fantastic, Crystal. Your comments about the incentive structures really are super interesting. So why are comorbidities not studied in depression, right? Because you want to know what fixes depression, and comorbidities are messy. Or why are the comorbidities not stopped in epilepsy? Because comorbidities make it messy. So if you're trying to publish a paper on how does epilepsy work, depression work, messiness, is problematic for that, right? And then on the other side, on the clinical side, I thought your research driving the screening for comorbidities was also really interesting, because when you're trying to treat epilepsy, you want to stop the seizures, and so you're focused on that. And so this hyper subspecialized world we live in, multidisciplinary care, is super important. And research and care look a lot like multidisciplines now, not just separate items. Quick question, and then a follow-up. The reward processing on epilepsy, you teased this. Do you have any preliminary data on it? Like, do they process slower because epilepsy?
Well, we think so. We think that it's taking longer. Like if their networks are not as honed or they're honed differently, it's just taking them longer to do it. But we are literally just starting to look at the brain data like this month on that. So we just finished the drift diffusion analysis, and we have some follow-up analysis to do before we take that measure into the brain and look at it. So it's coming, like as of starting later this month. But I don't have any data right now to show like the networks involved and why we think they're slower. So at learning or responding.
Just thinking about as a parent or a physician taking care of patients, then if your reward processing is slower, that has implications everywhere, right?
Yeah. Yeah, I mean, this is daily life pleasure and motivation, right? We talk about anhedonia, but anhedonic also means amotivation. It's not just the lack of pleasure, but the lack of seeking out pleasure. So getting out and doing things as much as like experiencing them well. I did want to comment on something that you mentioned, that like why do we exclude, like in depression, why do we exclude like neurological disorders? Because we believe that the depression signature, because it's signal above noise when you're looking at brain and behavior, right? That it's already kind of sensitive. And small, that we're not going to put in a lot of noise with a neurological disorder because we're trying to measure something that we think might be kind of small. So we need as clean a data as possible. But as we know, depression is super heterogeneous, as is epilepsy. So you have two very heterogeneous disorders. So it gets complicated really quick. But yeah, that's one reason why in depression we've excluded them, because we believe that the signal we're looking at is sensitive to those for sure. So, yeah.
What do you think this dialectic you were sort of talking about, the bidirectionality of function and anatomy? I thought was super interesting. So, for example, you said that depression causes decreased executive function, right? And so you're not able to plan as well. Now, does that bidirectionality mean that planning activities would have some effect on depression symptoms? What's the limits of that?
It's probably circular. We think that symptoms probably come from, like the symptoms that we see within depression or anxiety come from cognitive processes that are at deficit. Now, is it the chicken or the egg? Was the brain already doing it? Because as we know, what we do also changes our brain. Mm-hmm. The very cognitive processes that we engage in modify our brain. Why do we do cognitive behavioral therapies? Because it's supposed to help the brain. And so, but the brain is at a deficit causing these deficits, so it is kind of circular. So I can't say with confidence that executive function issues cause them versus do these come from somewhere and then it causes further executive function and things just sort of pile up like an onion. So
it seems like if you've identified these, then there's these therapies.
Yes.
Right, that could come from that, not just
right.
medicinal therapies, but behavioral therapies, cognitive therapies. Right. Testing therapies.
Yeah, or certain drugs that hit those networks, right, more globally or systemically. Like TMS and ketamine have been used to treat depression. We're now learning like what it's doing in the brain. TMS we know like DLPFC activity. You're trying to control top-down processing. You're trying to exercise that muscle so there's less bottom-up oversight rather than the top-down oversight of brain functions. Ketamine sort of like, I joke and say it's like going to a chiropractor and getting your back aligned. Like ketamine sort of just shuts everything down and everything starts to come back online. And like that's why people would come out of like surgery and be like, oh, I've never felt better in my life. It was sort of after the fact, like, oh, my depression's like gone. And that's when we start realizing ketamine actually changes mood. And so, but we don't know why. Now we're starting to learn why that is, and so. But the idea is to target, whether it's augmented therapy that's very targeted to frontal lobe function or making people go out and do cognitive behavioral things that improve hedonic experience, or certain medications that we could then target like whole systems.
I could keep going. We're going to keep us on time though.
Yeah.
Thank you so much,
Yeah.
Crystal.